Patricia and Le Studium, of course. It's interesting because I had the opportunity many times to present in Le Studium conferences, and also part of my story here in France starts with Le Studium, actually, and I'll tell you guys why. Exactly. But of course, my presentation today is a bit difficult because it's not exactly science. It's to discuss possible relationships and funding collab opportunities between Brazil and France. I could say from the start that I had not the best and most positive opinions about this because I could not get much funding between both countries. But most of my career path and time I put in science was exactly between Brazil and France. So what I present here is not an unbiased view on this potential relationship between the two countries in terms of science. So it's not unbiased at all. So it's just my opinion, and of course, to open discussion for different possibilities. What I leave while trying to create this collaborative efforts and what I've got, right? So I'll just share now my screen. You'll probably be able to see it right now.
We can
Is it
see
fine?
it.
Is it
Yes.
fine?
Yes.
Perfect, great. So that's what I would try to present to you today. So it's just a brief. I'll use my career path as an example to try to place things and discuss a bit on all the experiences I had with this collaboration. So I'm Brazilian. So as was already said, I'm from Minas Gerais. Uh, Look at those beautiful trees in Belo Horizonte. It's like I miss this a lot. So my graduation was in biology, essentially physiology and pharmacology, but essentially natural sciences. And during this period, as much of our Brazilian graduation students, we have the opportunity to go to the lab very early in our careers, more or less as we called it, the scientific initiation. So I had the opportunity to start very early my academic career in Belo Horizonte. It was by 2001. I've been funded through to funding coming from the CNPq and FAPEMIG, which were the essential, the strongest agencies in Brazil by that time, one of the strongest. The FAPEMIG was really good at that point in Brazil in 2000, in the early 2000s. I did my master's and my PhD, my thesis, in the same university in topics ranging from physiology to immunopharmacology. As I kept the same path, I stayed the 10 years I stayed at the University of Minas Gerais. I stayed in the same laboratory because I actually had no real reason to move to another laboratory because things were going very well. And I had several opportunities to develop good research. I wanted to follow this career path to be researching academia. And I choose to stay. This because Mauro Teixeira, my supervisor during all this time in Minas Gerais, was for a long time developing several types of study inflammation or host pathogen interactions. I was very keen to study this topic. And by 2008, 2007, 2008, Mauro got a lay studium fellowship to collaborate with Orléans, a city next here from Tours, and where the students based with Bernard Riffel, which is a now retired professor, or soon retired professor at the CNRS, a researcher at the CNRS in Orléans. But also with François Trottin, which is based in Pasteur Lille, someone I still have contact with, to develop things around trying to identify better the mechanisms involved, the immunological mechanisms involved in dengue virus infection, which by this time there was a growing interest. Vaccines were being developed. This was around 2008, 2009. It was being developed in the world. There were no exactly optimal vaccines by that time, no drugs available. The disease touches most part of the equator region of the world and then subtropical areas of the world. So that's a very serious concern. It's interesting because I could come here to study dengue, which was atypical by that time. This type of disease to be studied in France. Not much groups were studying that. People were studying the virus, but not developing essentially models. It's interesting, I say, because now we have the mosquito, the vector, well implanted in France. And we start to have like local cases of dengue in the south of France. So like 10 years, 12, 13 years ago, this was not yet a reality, but now it's becoming a reality. And I will come back to this aspects of changing, shifting the way of seeing things like later in the topic. But anyway, I could develop a research trying to identify cells and molecules involved in the immune response to dengue with the help of Bernard Riffet and François Trottin in France. So I spent my part of my PhD funded by the CNPq from Brazil as a sandwich PhD. And so this publication is a resume of things we've developed for around 7 years in the dengue topic between Brazil and France. I published this with Bernard a couple of years ago. It was by the end of our last articles were published on dengue. So it was important because with the mouse model we had in hands, we could show several things that later were shown to happen as well in humans. For example, the role of IL-17 and IL-22, two molecules related in the dialogue between the innate immune system and the adaptive immune system. Also about the iNKT cells, which was a topic from François Totin. It's a type of innate immune cells as well that were later shown to play a role as well as in humans. So of course, we did not found like a vaccine or a treatment, but we could show that a mouse model was correlating well to what is found in humans, which is most part of the time not the case. But we're quite happy with the research we developed at that point. And unfortunately, well, I changed topics of research, and as I show later, but we tried to fund a project in France by that time in a collaboration between the CNPq and CNRS, if I am not wrong, by 2009, 2010, but the project was not funded, especially because at that time, emerging viral diseases were not a hot topic in Europe. And we'll come back to this topic of emerging diseases later here in the topic. But in any case, it was a very prolific relationship and of course primed my research career with a strong French accent, which in the end, I would end up here, as you can see very well. So for my postdoc, it was essentially a natural course as well, because I studied a lot of cells and cellular immunity in the context of dengue. So I wanted to get more involved in mechanisms independent of the disease. So I shift my research to Glasgow. Eddie Liu, a professor at the University of Glasgow, and welcomed me in his laboratory for 3 years of postdoc after that research, which would develop a little bit of things on dengue as well, but also especially on mucosal immunity, notably mucosal in terms of like lung inflammation and intestinal inflammation. But of course, we did some dengue, which was prolific, and we could publish some work and still keep the collaboration with France alive by that time, since Bernard was involved in this project as well, since they were also longtime friends. So as Patricia mentioned to the students in the beginning, I mean, this is the importance of network as well, because all those guys that supervised me in this early career path were essentially friends of friends. So that's what I will not stick to hardcore science today, of course not, but essentially what I studied there, which is IL-33. It's a molecule, a cytokine produced during inflammation, which is involved in several mechanisms of diseases, from allergic diseases like asthma to infectious diseases as dengue, because it's an alarmine. It's a protein that is produced upon tissue injury, and in several diseases or infections, we have tissue injury. So the role of IL-33 is key because of that. Acting locally, it orchestrates different types of inflammatory responses composed by different types of cells, and also signaling through different types of signaling pathways, showing that it's a pleiotropic molecule with huge potential. So Eddie Liu created like a whole career around this protein, and I'm glad I had the opportunity to do this because it gave me a strong background on cellular immunity as well. So, and this was interesting because by that time I was already in a Franco-Brazilian relationship in terms of marital status, I'm saying, of course. And I saw this opportunity coming from Tours for someone was looking for immunopathologist with interest in studying mucosal a type of mucosal infection of chickens. And I said, why not? Because it was essentially what I was doing with mouse models, a bit of infection, a bit of mucosal immunity. And then Cathy Schuler had this open position at the our unity where Fotini works, where Patricia was welcome. So the Unity ISP, which is a big unit with like 500 people per maybe, I think something like that, and working essentially infectious disease of farm animals, mostly, or vectors or worms, but everything more or less related to farm animals most part of the time. So the chicken was obvious, an obvious species, target species to be studied there. And I accept that challenge that was to go deeper into the immunological mechanisms of this diseases, this disease, the avian colibacillosis, which is essentially a bacterial disease that is transported through air during poultry in poultry farms, during poultry commercial, even or backyard production. You can have colibacillosis. This is one of the, I think it's the most frequent bacterial disease of poultry, to not to say the least. And it causes a lot of death. It's complicated. The use of antibiotics is banned in several countries. So you cannot use just antibiotics. Vaccine is not that efficient. So people are still looking for things that are very efficient to avian colibacillosis. And most important, because as for most diseases, I mean, if we do not understand immunological and physiological mechanisms underlying the immune response of the host to the pathogen, it's very complicated to devise treatments or vaccines. And for chickens, that was not different. For most of you, I'm a biologist, but for veterinarians, know that, I mean, we know a lot of clinics, we can know a lot of histopathology, but we don't exactly know all the details to the same level as we know for mouse or humans, because, I mean, lack of interest, lack of money, because of course we need to do it, but the focus there is put in funding this type of research is not the same. And even if it's like we eat these animals, right? So pig and chickens. But anyway, I accept this challenge because of this as well. So to go into mechanisms that are, for me at that time, I could not understand why they were not known exactly. And that's what more or less what I developed in my first 5 years at the INRAE, which was essentially to identify how the bacteria enters in the respiratory tract, which cells will see this bacteria first, how they will respond. Is this bacteria going to the blood? If it's going to the blood, how is it going to the blood? But trying to characterize which cell types of the host could be the most interesting to, for example, if you want to immunostimulate the response. So many research groups in the world were, of course, trying to do the same. And I could quickly understand that most of us, which I call now us, we're having the same problems, which is essentially the development of new tools, because for the same reasons, I mean, it's a lot less interesting which is fair enough, but it's less interesting to finance and to produce antibodies or other molecules to farm animals instead of producing for humans and mouse, because you sell a lot more in general. And especially because you have less people asking for, which fortunately in the last 10 years, I could see a growing demand for new tools and reagents to work in farm animals, but it's still less interesting in terms of money to develop tools for this, which is changing, of course, in time. I also did a lot of mechanisms, fine mechanisms of trying to identify the host pathogen interactions in the chicken, using tools we developed in the lab, like for the qPCRs, gene expression, basic things, but the annotation in the genome of the chicken is not as good as the annotation we have today for mouse and humans, for example. So we still struggle sometimes to identify genes and stuff. So the same for the antibodies, the Western blots I did here, for example, with the signaling pathways. Most of those things that we are showing here are actually very new, not necessarily biological, because in other species things will happen more or less the same way, but it was the first time you're showing birds. Works more or less the same way it works in mammalians. So, and so anyway, I mean, it was important for me, and I think for people we're collaborating with in the veterinary immunology, comparative immunology domain, that we could do things similarly to what can be done in mouse or humans. We can have access or develop the same tools or the same reagents to work on the same level of technology, as we could show in a collaboration with the Roslin Institute, which is, of course, like a renowned institute on veterinary immunology, more specifically on veterinary science, to be more broad, to develop like lung explants to study host pathogen interactions in vitro, which, I mean, chickens, most of the diseases affecting chickens, the most serious are the respiratory ones. If you think, for example, about colibacillosis or avian influenza, though those are respiratory disease. And by that time, very recently, we didn't know, for example, which cells are present, immune cells or things like this are present in certain regions of the chicken lung, things that were known for humans or mouse models for years. So, and the other point at that time as well was that I was getting interested, and I get back to some political shift now, in studying the gut microbiota of these animals and possible correlations or relationships with the immune system. Why? Because the microbiota is not only a hot topic in human sciences or in the clinics for humans, but of course you can imagine for livestock animals as well. And especially for chickens, there is a huge interest in trying to boost a more robust, a more precocious, more an early and strong microbiota in early life, to try to push the immune system as well forward, especially because the most critical days for the young chick is like the first 7 days for infection or for having the development of an appropriate microbiota. Knowing that in Brazil or in France, the microbiota of chickens in the industry are relatively poor because they have a microbiota that's essentially the environment they live with. They're not acquiring the complex microbiota of the hens of their mothers, as would likely happen much better, like for example, with the humans. So we have people showing now very clear today that the starting, the triggering of the development of microbiota in chickens can be slow or poor if not enough beneficial bacteria are put in the beginning of their life. So, of course, you can imagine that the industry and as is as well much interested in this type of research. And what I would say is that the industry was more interested and more keen to dedicate funding to this type of research, at least in France, as compared to the academia. So we and colleagues get a much better feedback from the industry to develop topics like this, as compared to the academia. And this is pretty obvious, but it's not deleterious because I don't want to put the industry here as the good or the bad. That's not the point, but it's pretty fair to say that nowadays in a laboratory research in France, and especially working with livestock species, we are funded essentially almost 50-50, if you want, as compared to academia and the private sector. Because, I mean, for both sides, we need this exchange, the money, the exposition, the support that will be given to develop a product, for example, together with them, but also on the money necessary to develop the research we want, because there's actually no barrier all the time. Most part of the time, the industry wants you to collaborate with them and not necessarily give us the product. So this is another important aspect as well, but that I think in France is very well developed. And I think at the bureaucracy level, it can be complicated in terms of intellectual property and things like this, but it's well developed in France, especially with the INRAE. We manage to collaborate very well with industrial partners. And what is interesting as well is that those industrial partners, most of them are big companies with, for example, part of their people in Brazil, which I knew some of them, Filio, Agicio, and other companies who collaborate here in the poultry sciences. They have like pilot units in Brazil, close to producers. I could meet several Brazilians there, and it shows that there might be the possibility of strengthening this collaborations through exchanges. But there was something that was not something that I never addressed because I didn't need to in these industrial projects. But I know that this could be explored further. and not restrain only to academia. So it's important just to mention that those animals that we have here, for example, are germ-free chickens, chickens that have no microbiota at birth. So we can use these animals and compare to animals that were colonized by different microbiota and see what happens if you have no microbiota. And what happens, we know more or less, because you can see that several metabolites from the intestine are very differently compartmentalized. These animals have poor, absent, or almost very low levels of short chain fatty acids, which are very important to keep the dialogue between the microbiota and the immune system and even other physiological systems. And what is happening is that, as I mentioned, we try to push the at least for avian immunology, but also comparative and veterinary research, to try to push it further to give like a certain degree of mechanisms. That's what we are doing now. We're trying to correlate the metabolites that comes from the microbiota, which cells they are seeing in the chickens, which organs of the chicken they are accessing, because it's obvious. I'm not saying there's no spoiler here, but the industry might imagine, of course, to give things to chickens and wants the molecules coming from the microbiota to increase and do better things in the organs of chickens. So these people are trying to do this for years. But what there's a need, a societal need as well, to better characterize what we are giving to our animals, which could be in the case of vaccines or in the case of medicine therapies, but also for food and probiotics. So defining well which type of food or molecule we put in the food or probiotic we are giving to the animals, what is it exactly doing, which is good for an industrial, which will be happy to show that in details what their product's doing, but also to the population, which we know what we are dealing with, right? What you are eating at home. So Adiceo has been supporting us. It's an industrial partner, but also VetBionet, which is it's an European project from money from the European Union, support, like it gives us a lot of support to set this project. But again, this was not French funding. This was European, right? I will jump this. And then just to finalize, I mean, I've been involved in a couple of activities since I saw this need. I think by this time of my career, where things are scientifically developed, and the network in Europe is essentially well developed, but I still have this need of. I always try to have a better relationship with, like, for example, Brazil, but without funding it would be impossible to pay tickets, to pay the stay, and even if you have money to pay the experiments, that's not a problem. But you cannot just buy tickets from out of nowhere. So I mean, we need to formalize things to make this thing solid, especially to give good examples to other people. So we cannot just get money from the drawer and then pay for the people like this. I mean, you cannot do this in Europe. I know in Brazil you can do it, but this is wrong. We should not. But I unfortunately need to do it. But I try to, for example, I teach a couple of hours, not that much, at the Erasmus European Masters that welcomes several students, international students, and sometimes you have Brazilians. And I always try to get contact with these Brazilian students by the time I gave the courses, exchange contacts. And I was always, I mean, they always look for, they come to me, they just to chat. How is science in France? How do you work in France? And I find it very, very relevant, because nobody told me that to me, actually. By the time I came here, no one told me how things work in France, because Patricio, no, you tell me very well. I mean, it's another society. I mean, it's not another culture, right? So it's interesting to have this advice, or a more formal advice at some point when getting access, like, to this whole new culture and this whole different way of seeing science, right? And for another side, I try to contribute all the time with friends that often invite me to give an advice on a CNPK or a FAPEMIC project. It's less frequent now, but what was a lot a couple of years ago, at least put me in contact with this. But this was not enough, right? This is what I can offer as a Brazilian to my fellow Brazilians. But I tried then to very recently to formalize these things better, especially when I was with Katy Schuler working in bacteria. And we tried at least 2 formal collaborations to a Capes Coffee Cube project. And by that time, it was like in the early, I mean, 2016, 2017. And well, we tried. I mean, we discussed both topics, as I said in the beginning, are to study Escherichia coli, so the bacteria, the causative agent of colibacillosis, which is very important in Brazil and France. A lot of people is working on it in Brazil, to be honest. A lot less here in France because it's a mild problem compared to what happens in Brazil, which produces a lot more chicken than here. And two researchers from Brazil we got in contact during those times. One was Fabiana Horn from the University of Rio Grande do Sul, and also Rosilene from UFMG, where we came from in Belo Horizonte. So we have a really quick and nice contract. So we applied to the PHC project. And well, unfortunately, we didn't get it. With Fabiana, we didn't get it. Was proposing like this exchange of PhD students to work a bit with microbiology, hardcore microbiology in her lab, and coming here in France to work with cell biology in our lab. And with Rosilene, it was a bit different because Rosilene, we didn't get as well through the PhD, but by that time, was 2016, was one of the last waves of doutorado sanduíche, so the sandwich doctorate funded by the CNPq, if I'm not wrong. And she could come through this, so we paid for the experiments and for everything. She got the tickets, but we managed to. And it was a really great experience. I mean, we had the article published by the time Juliana left, so it was very quick. Juliana, the PhD student, the Rosilene, we managed to work with them. Unfortunately, could not make that type of research further. It was essential bacterial epidemiology, is not my topic. But as far as I know, it helped a lot for their lab in Brazil to establish on the topic. But anyway, I mean, there are several reasons that, to be honest, we never, as most of you know, we never get exactly what was really bad in the project. It was never very clear to us from Campus France and so on, like what was the real reason or proposal at that time. Or I can say that just because we are not happy, right? But anyway, we try to. And I think one of the triggers I had like in this approaches because what was the first time I was actively searching for money out of the human and mouse research, because I was not totally independent by the time I was doing human and mouse research. But with veterinary sciences, veterinary immunology, I was alone in a field that is not necessarily competitive, but where the amount of money is very low. So what triggered me is that at some point I say, come on, I mean, there might be something wrong, because people are not really interested in zoonotic agents and animals hosts, transmission hosts in contact with humans like wildlife and other stuff. People are not really interested in funding this, not possible. And then we had COVID. And it was appalling, the fact that, for example, 1 year before COVID, we got a refusal from another PhD with a colleague, where we proposed to study flaviviruses, not dengue, but different flaviviruses transmitted by mosquitoes, and to which the hosts were birds. And it was suggested like to be off topic. So they said it's much more ecology than human health or public health. So, and for us, it was not obviously not ecology. I mean, it was completely human health and immunology and everything, as most of us know now from COVID, the importance of identifying as well in the hosts, the intermediary hosts before humans, how the virus get there, how the host responds, which part of the host is involved in making the virus emerge. So all these aspects like caught a lot my attention. Now I can see that more projects are being funded on the topic. And another thing is that I could say as well that at least between France and Brazil, for the society I'm close to, like the immunology groups, there were not much interest as well for comparative and veterinary immunology. If you have a quick look at, for example, at the SBI, which is the Brazilian Society of Immunology, or the SFV, the French Society of Immunology, which sections are dedicated to anything outside human and mouse science, there is virtually nothing related to comparative immunology. Now we have COVID. There's a COVID section, but it's not an emerging disease section or well, I think you get the point. You can say that this should be covered by the virological societies or the veterinary societies, but of course they are not. I mean, the focus is other. I say that as well because, for example, in Europe, we have which are essentially Germans and the UK, which has strongly developing this networks in Europe on comparative or veterinary immunology, or both, through an European veterinary immunology workshop, which is held every 2 years, I think, in Europe. They have the German veterinary immunology group. The British Society for Immunology, one of the biggest in the world, has a comparative and veterinary immunology group, but in Brazil and in France we had nothing. And so it helps. Should I do something about it? I ask myself sometimes if I could be trying to. I try to have colleagues in the SBE, but this is not enough at some point. So I think the interest should come as well from us. That's what I try when I'm talking here, is because of it. But also try to identify Brazilian colleagues that want to try to put this into discussion in some societies that try to attract people, attract the focus, because if you have no people in focus, we essentially have no development in this topic. So just to finalize, there are like a certain initiatives that were taken, especially one that was very interesting, taken by the Brazilian consulate in Paris, which I could participate in 2021, which is the Diaspora Brasileira, so Brazilian expatriates living in France. I went there. We had a nice chat with people more or less with the same profile. Some people that had successful interactions, some others not. Topics that were seen the same manner by Brazil and France when building those fundings and structures like this. Some other topics that were often forgotten. So it was a very nice initiative. And I think we had another one in 2022. And so it's still going on. And then, of course, there are other guichets, other places to go look after it. Because, I mean, in the Embrapa, there is a huge collaboration with Brazil that could be probably better put forward by the authorities. Social media is very important, not only to offer jobs and potential open positions, but also to make publicly available what we are developing in livestock research. And that's it. That's what we are doing here today. So going to these places and those types of meeting to discuss the exchanges, where are the problems, that's the best we can do, like to engage in exchanges and dialogue with Brazilian universities and scientific societies. I think at this point, that's the best we can do, right? At least dialogue and exposing our problems, right? So I think that's it. That's it. I tried to cover a little bit of the story. Thank you very much. I hope you had fun.